I'll go as fast as I can, but I'm still going to do the material justice, okay?
Don't let us rush.
I can't be under that pressure. But
No pressure.
we'll definitely be done under our normal time. Okay, so let's finish up with our discussion of the gibberellins. We've been going through the different effects that gibberellins have on different parts of the plant. And so another thing that they do is they're involved in seed germination. In grass seeds particularly, there's this alurone layer.
He's not the only girl.
Yay! There's this particular layer called the alurone. It's a specialized layer of the endosperm that lies just beneath the seed coat. So if we look here at this wheat kernel, you see the endosperm here, and then there's the alurone layer right outside of that. And it's a protein-rich layer. So here's part of what happens during germination. As the seed is germinating, the embryo releases gibberellins. Those gibberellins diffuse towards the alurone layer. The embryo is down here, so it's the one that's going to release the gibberellins. But that gibberellin message reaches the alurone cells, which then produce hydrolytic enzymes. One of those hydrolytic enzymes is alpha amylase, and it hydrolyzes or digests the starch. So if you think about the benefit of that, this wheat kernel, which has sort of been in a state of arrested development as it's been in the ground, that embryo kind of awakens from its sleep. It causes this gibberellin message to be sent to the alurone, and now all that starchy endosperm is going to be digested, and it's going to fuel germination and growth of that young seed, that young seedling. So, as I said, that alpha amylase is going to digest all of those stored foods in the endosperm. All that starchy stuff is going to be converted to sugars, amino acids, nucleic acids. That's all going to be absorbed by the scutellum, right, by that specialized cotyledon, and then transported to the growing regions of the embryo. So I think this is kind of a cool little time-lapse thing here. Somehow they've gotten this seed with a transparent seed coat, and you can see the endosperm shrinking because of those digestive enzymes. Okay? If we just sort of look, this kind of says it all. Here's the embryo here. As it's germinating, it releases this gibberellic acid, which causes the alurone to release the hydrolytic enzymes, which digest the starches into sugars and amino acids, which then are absorbed by the scutellum, or this is that specialized cotyledon and monocots, and then that energy gets passed back to the embryo again. So that's that cycle there. And then finally, gibberellins cause bolting. Cabbages, carrots, other plants like that will form a rosette of leaves before they flower. Flowering is sort of, flowering is mediated by environmental cues, and flowering is usually induced by exposure to long days and or cold. All right, so cold is necessary prior to flowering as well. This is how the plant is orienting itself to the season. So it's got to pass through a cold season typically before it will flower. And then so following those long days or that cold exposure, the stems on these flowering plants will bolt. There'll be this sort of sudden elongation of the stems. And so we see a plant here that's bolted, this cabbage plant, it's bolted and then it flowers. So quick stem elongations and then flowering. But what scientists have discovered is that there's a gibberellin message there, as you might suspect. There's the cold season, and then when it's time to flower, gibberellins will be released, and then the plant will bolt, and then eventually flower. And the reason that we know this is that gibberellins can substitute for the light exposure, the long days, or the cold. You can just go right to the gibberellins, and you can cause bolting and then flowering. And so commercially, this can be used for early seed production. You can cause biennials to flower, usually they're gonna wait a whole year after that first year to flower. But once they go through that first year, you can cause them to flower and get seeds early if you give them gibberellins. Okay, so that's gibberellins, and then the last thing, they're also involved in the development of parthenocarpic fruits. We talked about this with the auxins. You can get some seedless fruits, right, with auxins, but some you can get through gibberellins, things like apples, currants, cucumbers, and eggplants, and some of these plants are ones that won't respond to the auxins. Mandarin oranges, almonds, peaches as well. So a prime example of this is that gibberellins are used to produce Thompson seedless grapes, a very common fruit that you would find in supermarkets around here, but these things have been given artificial gibberellin treatments. Okay. Hey, John. In order to get these seedless grapes. And so if you see here on the left, here are sort of these are Thompson grapes, produced the natural way. Look at how tight the clusters are. The grapes are a little bit smaller, they would have seeds. Whereas bypassing, making grapes the old-fashioned way, you treat the flowers with gibberellins, you can get these seedless grapes, which are looser clusters, larger fruits, no seeds. Okay? So the gibberellins, even though they tend to have this effect of causing things to happen, you go all the way back to the little fungus and it caused the stems to elongate. There are a couple of other things that they're involved in as well. Okay, so that sort of finishes that one. So moving into the last hormone lecture here. Up to this point, we've really just been talking about plant hormones in terms of the effects that they have, right? This hormone has this effect in this part of a plant. Okay, so it's just looking at overall effects. But I want us to talk now about the molecular mechanisms by which these hormones, these chemical regulators, are exerting their influence. How is it on a molecular level that hormones have an influence over growth or development or these other rapid responses that we find at the cellular level? How are they doing that? Okay, I want to talk about a couple of different things because it turns out that hormones are involved in some of these general areas. So we're going to talk about morphogenesis, and this is the development of a plant's overall form, the overall shape of an adult plant. The form of a plant, the shape of a plant, comes into being by way of a coordinated series of cell divisions and subsequent enlargement. So we'll talk about how hormones are influencing that. A lot of that's going to be based on the direction of cell division and the direction of cell elongation. And I've said this before, stem cells tend to divide this way and elongate that way, where leaf cells tend to divide this way and elongate that way. And some of that is under hormonal influence. We'll also talk about cell differentiation and how hormones are involved in that. Why is it that root cells look different and do very different jobs from leaf cells? They all came from the same zygote. They're all genetically identical, but it's because of the selective expression of particular genes within each individual cell. And we'll see how hormones are involved in that too, right? Hormones, as it turns out, will turn certain genes on and turn other genes off. All right? And so all of this, both morphogenesis and differentiation, are depend on good communication between cells, these multicellular plant bodies, and hormones, as we've already discovered, are playing a large part in this communication. So these hormones are doing the communicating, helping to coordinate growth and development by, as we've already said, acting as chemical messengers between cells. And I've already said this, but hormones can act as either stimulators or repressors. They're both activators and inhibitors. We can't think of them as one or the other. They're better thought of as chemical regulators or chemical messengers. And one of the reasons they're doing this is because they're stimulating and repressing specific genes in the nucleus. And most hormone responses are a result of this kind of differential gene expression. Okay, so we're going to talk about that side of things first. Even though I've talked about morphogenesis here and then differentiation, we're going to actually focus on the differentiation part first, and then we'll go back and hit the morphogenesis part. Not quite as much there with the morphogenesis. So that's where we're headed, that hormones are involved in differential gene expression, turning certain genes on and other genes off in cells. Now, there's a lot of evidence to support this idea, but one of those is the fact that plant hormones are influencing the rate of cell division and the rate and direction of cell expansion. This comment right here is actually tied more to the morphogenesis part, so we'll tuck that away. All right. This comment here is helping us to understand, setting us up for differential gene expression, because hormones are involved in that. This statement here is going to help us more in the morphogenesis part at the end. The fact that plant hormones influence overall plant shape because they influence the rate of cell division and the rate and direction of cell expansion. So differentiation and then morphogenesis. Okay, and so just as an overall, an overview that our chapter gives us, here are the 5 major hormone groups, and here's how they impact all those different things. Several of them have a positive effect on the rate of cell division. A couple of them have a negative effect that can inhibit cell division. Several of them have a positive effect on the rate of cell expansion. A couple of them have can have a negative effect. Here are sort of a survey of their impacts on the direction of cell expansion. Right, the auxins will cause longitudinal expansion, where we'll see later on ethylene cause a lateral expansion of cells. And gibberellins also cause a longitudinal cell expansion. And every single one of these 5 plant hormones has a positive effect on cell differentiation or gene expression. All right, so hormones are huge in both of these ways, influencing differentiation or selective gene expression and morphogenesis or overall plant shape. So 2 major ways that they're operating. Okay, so now with that general overview, we'll jump into the gene expression part more heavily. Okay, so you guys know that every gene that's in a zygote, right, in that very first cell of that little baby plant, every one of those genes is going to be in every cell of the adult plant body. All right, every cell in a plant body is genetically identical. One of the reasons we know this is because of what's called the totipotency of plant cells. That means that any individual plant cell has the potential to give rise to an entire plant. All right, because every cell has all of the same genetic information. But in any given cell, however, only certain genes are being expressed. That means that those genes are being transcribed into messenger RNA and then translated into polypeptides. And so the identity of an individual cell, not only the identity, but the function of an individual cell is determined by the specific proteins that it's producing. Okay? And sort of a general explanation for why that's the case, proteins, specifically enzymes, are catalyzing most of the cell's chemical reactions. So if you're producing this particular enzyme, then this particular reaction can occur within that cell. And then everything that flows from that reaction is possible in that cell. But in addition to that, proteins are also forming